I'm going to present our work recently accepted for publication concerning the behavioral and neuronal underpinnings of safety in numbers in fruit flies. I'd like to start off by giving you a brief overview of the background and motivation. Predation is thought to be a key driving force for group formation and social behavior, as being in a group can constitute an efficient anti-predatory strategy, allowing animals to benefit from a safety in numbers effect. When in a group, individuals can decrease their individual vigilance, display lowered stress responses to predator attacks, and finally also show decreased defense responses. Previous insightful studies have focused mostly on the ecological and evolutionary aspects of social behaviors within a group context. However, the underlying mechanisms by which the group exerts its effect on the individual are still understudied. This is precisely what we aim to tackle using Drosophila melanogaster, which in recent years has been shown to display quite varied and complex social behaviors. To study defense responses in fruit flies, we use looming stimuli, which reflect the way an approaching threat is experienced as an image that increases in size in the visual field on a collision course, and are known to elicit defensive responses in a wide range of organisms that use vision, including humans. In response to these stimuli, fruit flies, just like other animals, display flight or freeze responses, i.e. escape or tonic immobility behaviors. We use a monitor to provide 20 looming stimuli after a 5-minute baseline onto 2 arenas. Previous work in the lab had shown that in this context, individual flies respond sometimes with fast running escapes and mostly by freezing. In our study, I imaged the behavior of individuals and groups of up to 10 female flies and then analyzed the videos with dedicated software to determine each individual's behavior. Here is an abbreviated version of our results. First off, I'm showing you the fraction of flies freezing at 500 millisecond bins throughout the experiment, for flies tested individually in dark brown, all the way to flies tested in groups of 10 in yellow. The dashed lines represent the looming presentations. As you see, flies tested individually show the highest fraction of flies freezing. Flies in groups of 2 to 5 display an intermediate fraction of flies freezing, which reflect a lower time spent freezing per fly due to an increase in freezing breaks. Flies in groups of 6 to 10 display the lowest fraction of flies freezing, which was further due to a decrease in freezing entry. In summary, flies in groups display lower sustained freezing, an effect that scales with group size. For the next experiments, we focused on groups of 5 flies. To determine whether flies display synchronization of freezing entry, we tested single flies surrounded by 4 fly-sized manipulable magnets. For the experimental condition in green, after moving during the baseline, we froze the magnets from the onset of the first looming stimulus and observed the highest proportion of flies freezing. In grey are the controls, and I call your attention particularly to the light grey one, which is similar to the experimental condition but without looming stimuli presentation, and for which we observed the lowest proportion of flies freezing. These results suggest that flies use freezing by others as an alarm cue, which increases their propensity to freeze to an external threat, the looming stimulus. We then tested the role of motion cues of others perceived by a focal fly on freezing breaks by comparing groups of 5 wild-type flies in red with groups of 1 wild-type in blue and 4 blind flies in grey. Blind flies don't perceive the looming stimulus and walk for the duration of the experiment, so that during stimulation they produce a higher motion cue for the focal fly. In this case, we found that the fraction of focal flies freezing was lower than that of the control group, and that the reduction in freezing resulted from an increase in freezing breaks and a decrease in freezing entry. Hence, a focal fly surrounded by 4 blind flies behaves similarly to flies in groups of more than 6 individuals. We conclude that motion cues produced by others constitute a safety cue, since increasing social motion cues enhance the group effect, and therefore decrease the freezing responses of a focal fly. Finally, we tested the role of inactivating lobula columnar neurons 11, visual projection neurons that respond to small moving objects, using Kerr. In groups of 5 flies, this manipulation depicted in purple led to an increase in the fraction of flies freezing compared to control flies. This was due to the fact that each individual spent more time freezing as freezing breaks were decreased. In conclusion, diminishing the perception of the movement of others leads to a decrease of the group effect and thus an increase in freezing responses. We are really excited about our future research plans, as we now have a well-characterized conserved behavior that scales with group size, for which there are identified circuit-level entry points at the sensory processing side and at the motor implementation side. We can now take advantage of the existing plethora of tools in Drosophila melanogaster to uncover conserved mechanistic and circuit-level principles through which social cues affect individual behavior.